In this video, we're going to talk about cell respiration. So cell respiration is obviously this process here where the mitochondria takes organic molecules and oxygen and converts that energy in the organic molecules into the molecule of ATP, and it does that, again, to power cell activities, and it generates heat, waste heat in the process of that. So this diagram shows the movement of energy and matter through plants and animals. And here's a nice example of a mitochondria, the multi-layers of it, the inner membrane, the outer membrane, and the inner cristae, or the inner matrix within the cristae. So let's talk about first, why do we need ATP in cells? Well, again, ATP is the energy currency. So if you're involved in active transport and you're moving things in and out of cells, ATP will bind to and phosphorylate and sort of energize the proteins that are moving things out of a cell, so let's say for active transport, or moving things, contractual muscles, and things that move things in the cells, that ATP will bind to and phosphorylate a molecule and allow it to kind of contract and move. Or let's say we're synthesizing a big molecule from smaller molecules, that ATP will attach a phosphate group to one of the monomers and allow a covalent bond to be attached, again providing the energy to make that bond. So these are some examples of things that ATP does in the cell in terms of directly providing the energy or the power for cell activities. Okay, before we can get to ATP though, as we break down food, we actually are going to be in the process of shuttling these high energy reduced electrons, or electrons and reduced molecules, into other molecules, and they get carried to a final electron acceptor, which is oxygen, and in that process form water, a low energy waste product. So NAD is a electron shuttle. You can notice it's got a positive charge. What does that mean? That means it's missing an electron, right? It's one electron short. So you can see it's got the adenine and it's got the ribose and the adenine, so it's very similar to ATP, and it's got this unit here, and it's got this nitrogen here with the positive charge. So when it accepts these high energy electrons and hydrogens from food, it is then in a reduced form, and it's a high energy form. So it's basically carrying that energy, the high energy from those electrons from food, and passing it over finally to be accepted by oxygen in the final step of cell respiration. So we're going to talk a lot about how NADH gets reduced, or NAD gets reduced to form NADH, and carries these electrons and helps transfer this energy. So the first step of cell respiration is, first off, we're not in the mitochondria yet, we're out here in the cytoplasm. And it's a process whereby glucose is chopped in half. So glucose is 6 carbons. We chop it in half into 2 3-carbon molecules called pyruvate. And that process of splitting or lysing a sugar is called glycolysis. Again, glyco means sugar, lysis means splitting. So we're essentially just chopping it in half. And when we do that, we're going to use some of those high energy electrons in this high energy molecule to make 2-NADH from 2-NAD. Okay, we're also going to generate 2-ATP. Okay, and because we're getting that directly out of the glucose molecule, we call that substrate level phosphorylation, with the glucose being the substrate. So that's the first step in cell respiration. In the cytoplasm, enzymes essentially chop glucose in half into 2 pyruvates. We get a little bit of ATP, but then we're going to continue to get a lot more energy out of our glucose molecule. Here's another diagram, basically a summary of what happens in glycolysis. We've got 2 ATP that actually have to go in. We can call that like the activation energy needed to start the reaction. And when we put 2 in, we get 4 out. So notice we're netting a total of 2 ATP. If we put 2 in and get 4 out, we net 2. We're also getting 2-NADH out. Again, this is the high energy reduced form of this electron carrier. So we're getting 2 of these guys, and we're going to cash them in later. This is like, so we've got like chips in a casino. We're going to cash them in at the end and get money for our chips. But right now, we just have the NADH. And we also, of course, have the 2 pyruvate. Okay. So the next step, now we're entering the mitochondria. So this area in here is the mitochondrion. And we're going to chop our pyruvate, essentially knock off a carbon dioxide. And when we do that, we're going to attach this acetyl group on it, and this is called acetyl-CoA, where we attach it to this 2-carbon unit. And when we do that, we're getting another NADH, so another energy sort of carrier. It's going to transfer some of the energy, and again, we're producing another carbon dioxide, a waste carbon dioxide there. So this is sort of an intermediate step. We're moving the pyruvates into the inner matrix of the mitochondria, that inner kind of maze, and we're getting a little bit more NADH. Okay. So now here we are. We've got glycolysis. We generated 2 of these NADHs, these high energy carriers. We got 2 ATP out initially, and then we got 2 more NADH in the second step by chopping our 3-carbon units into 2 2-carbon units. Okay. Now we're going to feed these 2-carbon units into this cycle in here, and it's called the Krebs cycle. Whoops, the Krebs cycle. Okay. And when we do that, we essentially are chopping these carbons up and taking the rest of those high energy electrons on the hydrogens and using them to make a lot more NADH. So we're again oxidizing the molecule, the rest of the sugar, to reduce this molecule, and we're also reducing another molecule called FADH2. These both have big long fancy names, but they're basically B vitamins. They're the energy vitamins that give our cells energy, and they basically are just helping to transfer energy from food to ATP. So we got in that second step 2 more ATP generated and a bunch of carbon dioxide. So now we've completely chopped up our glucose into carbon dioxide, but we still have all these energy-rich molecules here. Okay, so this is a detailed look at the Krebs cycle, and you don't need to know any of this other than we're making a lot of NADH here and some ATP as well. And this cycle actually goes around twice. That's why we get 6 and 2 and 2 ATP there. And one thing is this, one of the intermediates here is called citrate. That's also known as citric acid. So again, just to summarize this, acetyl-CoA is chopped up into carbon dioxide. You can see the CO2s coming out here, and we're generating, the hydrogens are being used to reduce NAD into NADH, and we're also, again, the citrate is also called citric acid. So sometimes this cycle is called the citric acid cycle because of one of the intermediates there. Okay, but you don't need to know all of these details in this cycle. Just basically that we're chopping up a 2-carbon molecule into carbon dioxide, and we're using the energy-rich reduced hydrogens to reduce NAD and FAD into high-energy reduced forms of them. Okay, a simpler version looks a little bit like this, which just kind of shows carbon dioxide coming out, NADH is coming out, ATP generated, and then FADH2. Okay, so that's pretty much it, except we have this last important step here. So we have glycolysis, where we got 2 ATP, and we got then into the Krebs cycle, where we made 2 more ATP. But again, we also generated a lot of NADH. And then the last step, this is like we said, these are like your poker chips. We're going to cache these poker chips in this last step, the electron transport chain. And this is where we're going to get a lot of energy, right? How much energy? Well, a total of up to 34 ATP. So this is really where most of our energy is going to be extracted from the glucose. And this is a kind of a complex process. We're going to go into it a little bit more detail. So we're going to stop here, and in the next video we'll talk about the electron transport chain and how it takes that energy and oxidizes these molecules and generates all that energy to produce ATP.